Hello friends, today we are going to discuss one of the important topic, light reaction. That means photosystem 1 and photosystem 2. Light reaction. PS1 and PS2. Before going to the topic, we have to discuss about some of the important basics. Green plants and algae are considered as the producers, wherein the entire strata of consumers, the primary to tertiary, depends on the same. Why? Because they have the ability to prepare their own food material that are autotrophs through the process called photosynthesis. And what is photosynthesis? It's a physico-chemical process which utilizes the light energy to synthesize the organic compounds, and through which, and it is a basic food on planet Earth, and secondarily it is a source of oxygen generation which the entire biosphere has to depend upon. Okay, because of this, that means the entire food chain, the say food web, depends on producers. Apart from clean plants and algae, photosynthetic bacteria also have the photosynthetic ability, and this photosynthesis takes place in chloroplast, and it takes place in two distinct phases called light reaction and dark reaction. For light, there are two phases of photosynthesis. The light reaction and the dark reactions. Here, light reactions, the basic elements are photosystem 1 and photosystem 2. I told you, the synthesis of organic compounds, say the carbohydrate, by entrapping light energy, say solar energy, through the utilization of the raw material, that in the carbon dioxide and water, we prepare the food material, that in the carbohydrates. It's called photosynthesis. Say in the form of a chemical reaction, basic equation we knew that the 6CO2 plus 12H2O into sublight C6H12O6 plus 6O2 plus 6H2O. Here oxygen is coming from the splitting of water molecules. By splitting the water molecule, we generate oxygen, and the organic compound, that means the glucose, is the food material. That means the carbon dioxide and water are the raw materials, and we entrap the light energy, so to prepare the carbohydrates. So it's a basic reaction. It's all about basics. But the thing is, light reaction and dark reaction. Light reaction, the name it implied, the process has to be dealt under the presence of light, to the driving of light energy. What is dark reaction? Is it means that there is no availability or utilization of light, or it doesn't depend on the light? Conventionally we used to think like that, but it's a wrong construing. What exactly the dark reaction means? It doesn't depend on the light directly, but depends on the products of the light reaction. What are the products of light reaction? NADPH and ATP. Nicotinamide adenine dinucleotide phosphate hydrogen, the reduced form of NADP plus and ATP. These two have been utilized in the dark reaction, what we call carbon fixation reaction. Dark reaction, otherwise called carbon fixation reaction, so as to utilize, that's why to synthesize the glucose, that means the carbohydrates. That means dark reaction, it does not mean it takes place in the absence of light, but it means it may not directly depend on the light, but depends on the products of light reaction, that means the NADPH and the ATP, to prepare the carbohydrates. Light reaction means, say it implied, it's a physico-chemical process to synthesize NADPH and ATP through the triggering, through the driving of or through the utilization of light energy. Here the crux point or the central elements of light reactions are photosystem 1 and photosystem 2. They are the operating system. That means they are called biological machinery to execute the light reaction so as to get the NADPH and ATP. Okay, it's about the basics. Okay, and how the light reaction takes place. As I told you, it's about basics, the photosynthesis will take place in chloroplast. Again, there is one more component. In chloroplast, just like mitochondria, it's a double wall membrane. Inside is called stroma. Here, a flattened membranous sacs called thylakoids, which are arranged in the form of stock of pile of coins, known as grana. And these are the thylakoids which contains chlorophyll. That means chlorophyll pigment present in thylakoids, and here light reaction will take place. Whereas in dark reaction will takes place in the stroma. That means the place of occurrence in chloroplast is also different. It is a clear-cut example of balkanization, what we call division of labor. One part, say membranous part of chloroplast, say for instance the thylakoid membrane, will undertake the light reaction, but the stroma, just like the matrix, it is an open space present inside to the inner membrane of the chloroplast, will take care of dark reactions. Okay, say how it works. So for understanding, I'm giving a small issue. Close the chloroplast. It's a double wall membrane. And these are the thylakoids. And there is stroma lamellae, which connects different grana. For emphasis, I am only magnifying two issues, okay, two particular components. These are thylakoids. That means grana. And these are stroma. And that is stroma lamellae. In thylakoids, light reaction will take place because chlorophyll pigment is present here. In stroma, the dark reaction will take place. That means much specifically, thylakoid membranes is a site for light reaction. So just I would like to magnify the same. Say take this as a particular example. Now how it went. Say with respect to basic elements, the light reaction, as I told you, there are two elements, the photosystem 1 and photosystem 2. Again it have two subcomponents in each. One is called antennae or LHC, otherwise called reaction, other thing is called reaction center. That means photosynthetic reaction center. Same component. Antennae or light harvesting complex and reaction center. PS1 can also designate it as P700, whereas for PS2 it is called P680. I will tell you what exactly it means, okay, later. So it is about the basic thing, and we'll draw on how the issue ends. Say these are the antennae, say for understanding. This photosystem 1, and these are the axillary pigments. This is the reaction center, which contains P680. It is PS2. PS2. Then it is a primary electron acceptor. Then it had a series of electron transport chains. Electron acceptor, what we call now? Plastoquinone. Plastoquinone. Then cytochrome B6 F complex, then plastocyanin, simply PC, plastocyanin, then the photosystem 2, this is the reaction center here. It is called P700 reaction center, and these are different auxiliary pigments. I will tell you what exactly it is. Then this is the primary electron acceptor, primary electron acceptor. Then finally ferredoxin, then it leads to, for understanding I am drawing here, NADP plus leads to NADPH. Here the reaction will be like ADP plus PI to ATP. And these are the auxiliary pigments. That means, say, chlorophyll B, xanthophylls, phycobilins, etc. And it contains chlorophyll A. See, chlorophyll A. Here also chlorophyll A. And this is very important, the sun. Leads to, say, photons. Then here, then here, then here, then here for H2O, that leads to 2H plus 2 electrons plus half O2. Electron source, and this is photon. Okay, this is a basic schematic representation of photosystem 1 and photosystem 2. Let me discuss first of all. Photons are the basic units or elements in the light. They are nothing but the bundle of electromagnetic energy, sometimes referred to as quantum of electromagnetic energy, and they are the sources of energies. Say, how I don't know how the light energy has been utilized from the sun, for example, in general, and the same has been utilized for the synthesis of carbohydrate to the utilization of carbon dioxide and water. It is about an overall overview of photosynthesis and how the light energy utilized. Whether the light energy is like unanimous, that means if each and every wavelength of light has been utilized, that means if particularly any kind of wavelength, not exactly the same. There are favorite zones in the light. For example, we knew that visible spectrum, that consists of 380 to 760 nanometers, otherwise called VIBGR. Here the point is, before we're going to talk about, we have to learn about this. VIBGR means we have 7 colors. Whether all light, that means all wavelengths present in the visible spectrum of the light will be absorbed by the photosystems? No, not exactly. There is called heterogeneity. What we call favorite zones. Here the favorite zone is called red. Says somewhere around 680 to 700. Red is called favorite zone. That means absorption of light. Specifically means that interpretation of the absorption of light means absorbing the red light. That means wavelength of somewhere around 680 to 700 nanometers. It does not mean it cannot absorb the wavelength of other lights. It do absorb violet, indigo, blue, but except green. That is why that means the plants gives off the green color. That is why the plants appear in green color. It can absorb any wavelength, but red is called the most hot spot. By utilizing the same, it can have, it is efficiency of absorption, what we call, that leads to probable photosynthesis. That means what we call a appropriate photosynthesis process because of absorption of wavelength between 680 to 700 nanometers. That is why here P680 means absorbing the visible spectrum, that means the red region at 680 nanometers, whereas 700 with respect to PS1. That is why PS1 is otherwise called, is present in the form of P700. That means chlorophyll A pigment can absorb the light in the visible spectrum at 700 nm. That means red. In case of 680, it's all the same red. This is about the basic. Then how we will start? And I told you, photon is a high energy molecule, high energy component. This is the basic brick of light. And when this photon, I told you, the PS, that in both photosystem contain, there are two components: LHC, light harvesting complex, otherwise called antenna, and the reaction center. The light harvesting complex, these are all called light harvesting complex, made up of pigments, say chlorophyll B, that means accessory pigments are the crux point for light harvesting complex. The name it implies, it harvests the light, the first receiving zone of light. It absorbs the light, it absorbs the photon. Say the photon come here, it bounces among the chlorophyll pigments like this, and reach the reaction center. Just like, what we call resonance electronic transfer, or simply RET, resonance energy transfer. That means the photon has been transferred to the mechanism of resonance energy transfer mechanism. That means the chlorophyll P pigment, xanthophylls, phycobilins, these are called accessory pigments, and they constitute the LHC, that means light harvesting complex, and the antenna, which is the first component of both photosystem 1 and photosystem 2. It is the same. There are no difference. Only one difference in with respect to reaction center, I will tell you. Okay, first the same. That means the antenna or the LHC, they receive the light in the photon, and then it bounces off and reaches the reaction center, RC. With respect to photosystem 1, here what is photosystem 2 and 1 is the order of discovery. It is the order of discovery. It's not the sequence of chemical reaction. It's about order of discovery. Here the photosystem 2 is the first component. Okay, with respect to non-cyclic, I will tell you what exactly non-cyclic and cyclic. So the first component here is photosystem 2. That means P680. Somewhat we reach the reaction center. Reaction center contains chlorophyll A pigment. Here chlorophyll A pigment is a major chlorophyll that involves in the photosynthesis process. The rest pigments are called accessory. They will help the chlorophyll pigment, but here chlorophyll pigment absorbs the photon at 680 nanometer with respect to PS2. This is very important. 680 nanometer with respect to PS2. That is why it is called a reaction center. It absorbs at specific wavelength of 680 nanometers with respect to photosystem 1. Then the most important point, it has to, why they absorb the photon for excitation. For excitation, excitation of electron. How and what is the source of electron here? The water. I told you already. H2O, it leads to 2 protons and 2 electrons and 1 oxygen atom. And these two first electron, they will go in a line. These electron, when it reaches the photo reaction center, that means chlorophyll A at P680, that means wavelength absorption P680, and the photon will excite so that it will reach to the, that means we go upstream. That means not the downstream but upstream so as to reach the primary electron acceptor. That means after the reaction center, the electron goes to the excited electron to reach the primary acceptor, and after that it will enter into the electron transport chain, just like we discussed in the respiration system. That means like oxidative phosphorylation system we have discussed about. The same here, but this is a slight difference. The first component is plastoquinone. A set of respiratory, that means protein complexes will involve in the electron transport system. That means electron transport system 1. Here the electron will move downstream, not upstream, downstream. That means here the sequence of these electron carriers, that means plastoquinone, cytochrome B6F and plastocyanin, is with respect to the scale of redox potential, or what we call oxidative redox potential. With according to the same, they have been arranged. It will move down, so that means as it moves downward, the energy will be lower. This is very important. First we will reach the plastophenol, then the cytochrome B6F, then the plastocyanin, then it will reach the photosystem 1. Here cytochrome B6 is important. It is called proton pump. It is called proton pump. Just like in case of electron transport system, the proton, that means the ATP is synthesized, what we call chemiosmotic hypothesis. Here, synthesis of ATP is connected to proton gradient. Here, the only difference between here, that means the synthesis of ATP in chloroplast instead of mitochondria, here is the concept is same. That means proton gradient driven, but there the protons has been accumulating in the intermitochondrial space in case of mitochondria, but here they will accumulate in the lumen. That means inside of the thylakoid membrane is called lumen. That means collecting the protons from the stroma will, that means the proton from the stroma will get inside the lumen. That's the only difference. And that how they will synthesize the ATP. I will tell you how. That means since electron transport, the what we call balance of electron ionic balance. See, just we can understand like that. For electron transport, then the protons from here to stroma, they reach the inside the thylakoid. That means the lumen. Then during this process, the ATP will be synthesized. What that means? Cytochrome B6F complex is called proton pump, and this electron ultimately is the photosystem 1. That means the reaction center which contains chlorophyll A, which can absorb light at P700. That means it can absorb light at 700 nanometers. That means same red light. Here, so in case of the distinguishing feature between photosystem 2 and 1 is that it's with respect to reaction center. Both contain chlorophyll A, but chlorophyll A in photosystem 2 absorb the light at P680 nanometer, whereas in case of photosystem 1 it is 700 nanometers. That is the most important point. And after it reaches the reaction center, the same concept, the sun, say the photon reaches here, concept is same. The photon will bounces across along the pigments and somewhat reach the reaction center and reach the primary electron acceptor. And here the ferridoxin, the second electron transport system, we can say like that. And this ferridoxin, finally it goes to that means the electron along this finally goes to NADP plus, which is the final electron acceptor. That means NADP plus is oxidized form, it will reduce to NADPH. So here throughout this process we have synthesized two things, what we call potentially high energy chemical intermediates, ATP and NADPH. These are the products of light reaction. How? Say the first concept is same. The photon, they will enters into the antennae or light harvesting complex of photosystem 2. That means say here along it bounces off and the reaction center contains P680, that means chlorophyll A, which can absorb at 680 nanometers, then reach the primary acceptor, then into the electron transport chain, that means plastophenol, then the cytochrome B6F, then the plastocyanin. This is called Z scheme. They are considered as Z scheme because of its appearance. I told you the sequence is with respect to oxidation reduction potential. Simply redox potential. This is a parameter. What is the basis for the arrangement is redox potential, and that appear in the form of a Z scheme, that is called. Okay. Then it will have ultimately the photosystem 1. That means the reaction center which contains chlorophyll A, which can absorb wavelength at 700 nanometer. Then the same primary electron acceptor, the ferridoxin, the second electron transport, then the NADP plus become NADPH. During this process, the cytochrome B6F is called proton pump. That means ATP synthase. They will synthesize the ATP just through mechanism of chemiosmotic hypothesis. That means synthesis of ATP through proton gradient. The same in case of respiration in case of mitochondria, the same in case of photosynthesis of chloroplast. Except the accumulation of proton takes place here. That means the stroma, lumen of the thylakoid there in case of intermitochondrial space. That's the distinction. The concept is same. That is why phosphorylation, it is called photophosphorylation in respect to chloroplast. There it is called oxidative phosphorylation. That means mitochondria. That means phosphorylation driving by the oxidation. Phosphorylation in the presence of light. It is called photophosphorylation. Okay, the concept is same. and this entire scheme is called, that means whatever we say is takes place in a linear arrangement. Linear, that means in one particular direction, say here and here up to an NADPH. It is called non-cyclic photophosphorylation. Non-cyclic photophosphorylation. That means, here in case of Krebs cycle, we have discussed that Krebs cycle means it again come back to our starting point. Here there is no such thing, non-cyclic photophosphorylation. The products of non-cyclic, the entire scheme is considered as non-cyclic photophosphorylation. The two products, ATP and NADPH. It involved both photosystem 2 and photosystem 1. It takes place in thalochromate membrane, but sometimes there is a possibility of cyclic photophosphorylation, where the cyclic photophosphorylation doesn't take place in thalochromate membrane, but it takes place in the stroma lamellae. What we call cyclic photophosphorylation takes place in stroma lamellae. Why? Because there is no PS2 and NADP reductase. Lack of availability of NADP reductase which can synthesize the NADPH, and no PS2. In stroma lamella, I told you, stroma lamella means that connects different grana, that means different thylakoids, different grana. There the cyclic photophosphorylation takes place. It means that what exactly is cyclic photophosphorylation? Only PS1 is involved here. Say this particular component will be involved. No PS2. What exactly happens, Say this is reaction center. Instead of going to the pyridoxine, it will again come back by means of electron transport chain. That means the cytochrome B6, then the plasmocyanin, then again the reaction center. And during this process, the ATP is synthesized. The concept is same. The only thing is it bypasses the photosystem 2. That's it. No photosystem 2 and of course obviously no NADPH. Only thing is here photosystem 1 is involved in case of cyclic photophosphorylation. So only ATP is produced here. Only ATP. So the distinguishing feature between the cyclic photophosphorylation and non-cyclic photophosphorylation is, in case of cyclic photophosphorylation, which it involves only photosystem 1, because it doesn't involve, it contains availability of photosystem 2 and also NADP reductase, and it takes place in stroma lamellae, and it only synthesizes ATP. Whereas in case of non-cyclic, the entire, in general, it is a non-cyclic photophosphorylation because it is a linear arrangement. Say photosystem 2, the electron transport chain, and photosystem 1, then NADPH. That means both ATP and NADPH are produced non-cyclic. Obviously, it produces ATP and NADPH, and it contains, it involves both photosystem 1 and photosystem 2, and it takes place in thalochromate membrane. So it deposits the most important things is, in case of photosystem 1, the P6700, which absorbs wavelength at 700 nanometers. In case of photosystem 2, that means chlorophyll A, P680, which can absorb the photon at 680 nanometers. In case of cyclic, only ATP is produced. In case of non-cyclic, both ATP and NADP is produced. In case of cyclic, no photosystem 2. In case of non-cyclic, both involved. Okay, it's about light reaction, no? And the products, that means NADPH and ATP, will be utilized in the dark reaction, that means the stroma of the chloroplast to synthesize carbohydrates. We'll discuss tomorrow. Okay, you may like, share and subscribe to it.